Hello everyone, and welcome to this Integrated DNA Technologies webinar on getting started with CRISPR genome editing. My name is Dr. Hans Packard, and I'll be serving as the moderator for today's presentation. The presentation today will be given by Justin Barr. Justin is the product manager of functional genomics at IDT, where he works closely with researchers to develop new tools for improving molecular biology research. Justin has a bachelor's in science from the University of Iowa, with an emphasis in genetics and biotechnology. And prior to joining IDT, Justin was a part of a research group at the university where he studied effects of VWF and ADAMTS13 on endothelial cell dysfunction. Justin's presentation today should last about 40 minutes, and following that presentation, we'll take as many questions as we can from the audience. As attendees, you have been muted, but we encourage you to ask your questions at any time or make comments at any time during or after the presentation, and you can do that by entering them into the questions box in the GoToWebinar control panel, and you'll find that at the right-hand side of your screen. And there's a little down arrow. You can click on that and make the window larger and type your question into there. And there's actually a thing too on the right-hand side of the bar in the newer interface where you can just pop that window out and type your question in. Makes it a little easier to type in that larger space. At the end of the presentation, then, I will pass as many of those along to Justin as I can. We will get through quite a few of them. If we don't get to your question, we will follow up with you by email, so please make sure that you ask your questions and you will get a response from us. Also, in case you need to leave early today or you want to revisit this webinar, we are recording the presentation, and we'll make that recording available on our Vimeo channel and our YouTube channel, which is shown on the screen. And it will also be on our website. We have a video library under our support and education tab, so idtdna.com, and we have a whole video library. There's a host of CRISPR webinars already there that address other areas of CRISPR research, and we also have a variety of other topics on NGS and qPCR and a whole host of other molecular biology topics. We also get a lot of questions about slides. The slide deck will be posted. It's actually already posted at the SlideShare site that is shown, and we'll also be sending you a link to all of this stuff in a follow-up email. So, with all of that being said, I'm going to hand this over to Justin now, and Justin can get started.
Great. Thanks so much, Hans, and thanks to everybody for joining the webinar today. I'm excited to be talking about how to get started with CRISPR. We'll be reviewing gene knockout and a little bit about homology-directed repair as well. My goal for this webinar is to really provide what I hope to be an easy introduction to designing your guide RNA for CRISPR, and then also to give you some sense of what you might need if you want to jump straight into homology-directed repair. So if you don't have a CRISPR background already, that's okay. We'll touch on the basics as I go through, and then as Hans mentioned, I'm happy to answer questions at the end. So to get started, quick overview of what I'd like to cover. First, a basic CRISPR workflow to give us some background to consider as we move through the rest of the steps. And then I'll actually jump straight into planning for a knockout experiment. So if your goal is to disrupt gene function and perhaps assess the impact of that gene, we'll talk about designing a guide RNA to actually disrupt that gene. We'll move into some discussion of delivery methods and comparing things like liposuction and electroporation. And then at the end, I'll spend a few minutes talking about homology-directed repair templates. Things to consider, and then how you would actually go about designing and ordering that type of repair template. So this slide provides an overview of a kind of current CRISPR workflow if you're working with CRISPR Cas9 protein itself and a guide RNA. That's the approach that we really like to use, and our ALTAR system is based around delivering the Cas9 protein along with a chemically synthesized guide RNA. Now, the goal here is to induce a double-strand break at a specific site within the DNA of your cells. And the way this is accomplished is by first complexing together a guide RNA that tells the Cas9 protein specifically where to go and where to induce a double-strand break. Once these two are complexed, they form a ribonucleoprotein complex, that has a 20 base specific sequence as part of one of the 2 RNAs that are used, and that 20 base sequence provides the specificity to the target DNA such that ideally only in those locations a double-strand break occurs. For Cas9 nuclease, which is what I'll be referring to today, that's typically a 20 base protospacer sequence, and those 20 bases are immediately upstream of a motif NGG. That's commonly referred to as the PAM site, or protospacer adjacent motif. And that NGG sequence always has to be present in the target just downstream of the 20 base target DNA, so that the Cas9 molecule will know to stop and interrogate whether or not it has a 20 base match with its guide RNA. Assuming that it does have a match, the Cas9 RNP will induce a double-strand break in the wild-type version of the protein. That cleavage event happens between the 17th and 18th base of the target DNA, and it is a blunt-ended cut, so there's no overhang left. Once that double-strand break has occurred, then we either rely on a non-homologous end joining process, and that's the more error-prone process where typically an insertion or a deletion event can occur, or we rely upon homology-directed repair, where a template piece of DNA is used to align and repair either as an error-free process, or if you're introducing a specific mutation, that can sometimes be included in the middle of that repair template, and we'll talk about that towards the end of the presentation. So getting started with CRISPR, if this is your first CRISPR experiment coming up, it can be a bit overwhelming because we have so many different ways that you can actually implement CRISPR-Cas9 genome editing. Overall, the goal is to be able to get Cas9 protein, a guide RNA, to complex together into that ribonucleoprotein complex. But getting to that point can be done through a number of different methods, whether that's delivering those, either the Cas9 or the guide RNA in a plasmid form as DNA, or as linear DNA, such as a GBLOX gene fragment, or perhaps delivering those components as mRNA or a transcribed single guide RNA. All of these methods can work very well. There are advantages and disadvantages to all of them, but to really narrow this down and simplify the process, we feel very confident that the best way to deliver CRISPR reagents is just to work with the closest to finished products as you can. So that means working with delivering the functional Cas9 protein along with a functional and ready-to-use guide RNA. This allows you to actually pre-complex these two and form your Cas9 ribonucleoprotein complex before it's even delivered to the cells. And so that allows you to have an active complex, put it into cells, and it can be immediately active to induce double-strand breaks within those cells. So this is the method that we'll be using as I explain further delivery methods and approaches in the rest of this webinar. So taking a look at the basic workflow for a CRISPR experiment, I've kind of broken it down into 5 distinct steps here. So it begins by designing your guide RNAs, so figuring out specifically where in the genome you'd like to target, and then how to identify which 20 base sequence is what you'd like to use to try to target and induce that double-strand break. Once you've designed those guide RNAs, then you need to order and prepare your guide RNAs and complex them together with the Cas9 protein. So you actually assemble that ribonucleoprotein complex. From there, you're ready to move into the delivery stage, which typically is done either through liposuction, electroporation, or microinjection into cells. And then finally, after the CRISPR delivery has occurred and some time has elapsed for that editing to take place, the genomic DNA is collected from those samples and you move on to analysis. And we'll talk about each of these steps in a little bit more detail. So starting with the guide RNA design. We'll take a look at that first for a gene disruption knockout type experiment, where the goal here is to select a guide RNA that matches a coding sequence for a gene of interest and induces a double-strand break within, say, an exon of that gene. And then we use the non-homologous end joining process and the error-prone nature of that process to create an insertion or deletion event, ideally disrupting the function of the gene, either through knocking the, causing a frame shift mutation, or perhaps introducing a stop codon prematurely. So the first step is to actually identify what type of design tool you're interested in using. If you type CRISPR design just into an online search, you'll find that there are a lot of different tools out there. And it's great to have a lot of choices because each of these has unique advantages or disadvantages. And so I want to point out a couple important considerations to think about as you choose what type of tool you use for your actual experimental design. All of these are good tools. I don't really wouldn't avoid any of them, but it's important to think about what type of species or cell line was used to set up that tool. What if it's based off of an algorithm from experimental data, how does that compare to the type of experiment you're doing? Were all the experiments run on human cell lines and you're using a mouse or perhaps a zebrafish experiment? And in addition, how was the Cas9 and the guide RNA actually delivered to those cells? Many of the algorithms developed have been based off of a lentiviral delivery system, which can be very effective and a great solution for high throughput CRISPR analysis. But when you switch from that type of delivery method to a DNA or direct RNA delivery of a guide, or even a ribonucleoprotein delivery of Cas9, we actually see that there isn't always strong correlation between the guide RNA design and what's effective in one system versus another. So the source of these components and the method for delivery can make a big difference in how effective a particular guide sequence might be. And finally, all of these tools will have some kind of design output, and many of them provide both an off-target score and an on-target score, corresponding to how likely this guide RNA sequence is to cut somewhere unwanted in the genome, and then how likely this particular guide RNA sequence is to cut efficiently at the intended target site. Now, sometimes you can see both scores and rank them based on either metric as you'd like, and sometimes these are actually weighted together, and it's important to understand what that weighting factor is so that you can decide which of these factors is most important to you for the particular experiment you're working on. Lastly, some of the tools will also filter out guide RNA sequences, either due to polybase runs or some other metric. And typically, how that filtering is done is pretty transparent, but it's important to have some idea of how the tool was designed and whether or not it matches well with the experiment that you're trying to do. So I'm going to go through two different examples, take a look at two different free, easy-to-use tools that are online for choosing guide RNAs. The first one we'll take a look at is based off of a custom track on the UCSC genome browser. So a very commonly used tool. The nice thing about this is this custom track already has all of the guide RNA sequences designed, and you can simply look at the gene sequence you're interested in and determine if any of the particular guides that are shown will fit well for what you're trying to do. We'll also take a look at the CRISPR design tool from MIT. This one has been around for quite some time, and while it doesn't provide an on-target CRISPR analysis or ranking, it does a very nice job of providing a simple output for off-target analysis. And so this is another common tool, and we'll take a walk through that tool as well. The latter tool is very convenient if you don't have a commonly used genome. So the genome browser track only works for a number of species, and sometimes you can use the MIT tool for an even wider number of particular species. So to go ahead and jump right into some use of these tools, I'll begin with the genome browser, which is simply genome.ucsc.edu. And I already have the home page pulled up here, but what I'll do is jump straight into the human genome track. We'll go ahead and pull up the latest assembly of the human genome. And I should have the default view here. So if you don't regularly use this genome browser, your screen should look a lot like this when you pull it up for the first time. Lots of information, much more than we need for right now. So to simplify things, I'm simply going to use the Hide All button that eliminates all of the custom tracks that have been added right now. So I'll hide everything, and I'm going to turn on just two different tracks. The first one is the NCBI reference sequence track, and the second one is this CRISPR track, and that's the one that will actually show us all the guide RNA sequences that are possible. And so now if I refresh, I can see that I have both of those tracks present along with my location, in this case on chromosome 7. So just as an example, let's try to find a guide RNA sequence that would work well for disrupting the beta-actin gene in this human genome. So we can type in ACT-B here, and we're looking for this first gene target. And I can simply click Go. And now I see the entire beta-actin gene. In this case, based on the arrows, it's being transcribed from right to left as I look at it. And I can see both the exons and the introns of this gene. So in choosing a location for your guide RNA, if I'm trying to disrupt a gene, I generally choose or start with exons that are earlier in that transcript, just so that if I'm causing a disruption in that protein, it happens earlier in the coding sequence and is more likely to lead to a non-functional protein. It just makes it a little bit easier to analyze the results and make sure you don't have a partially functional protein that isn't actually accomplishing what you're trying to do. So in this case, we'll take a look at this early exon here, and I can just drag a box over the scale bar, and then it'll allow me to zoom in on just that exon. So now you can see the CRISPR track in a little bit better detail. And what this track is showing is a color-coded scheme of all of the guide RNAs that exist within this region of the genome. So anywhere where there's an NGG sequence, it tests the 20 bases upstream of that PAM sequence. And for all of the 20 base sequences in grayscale, the specificity score is below 50 on a scale of 1 to 100. And generally that's a little bit more worrying. So it has some off-target potential. It might actually cut somewhere else in the genome. And unless there are no other options, I typically would avoid such a guide RNA. For the guide RNAs that are colored, we can see we have green, yellow, and there's one red one here in the corner. Those all have specificity scores above 50. And then the color represents the on-target editing score. So basically they're considered safer, and then we move from higher scores for on-target to kind of moderate, and then this red one is a lower score, something below 30 for the on-target cutting efficiency. So the nice thing about this custom track is it allows you to very quickly see all of the potential guide RNAs, quickly determine which ones might be predicted to be better. And then if you're using certain browsers, I'm using Chrome right now, you can actually hover your cursor over each guide RNA and it will tell you the specific scores for, in this case, 3 different algorithms that are being used. The first is the MIT specificity score, which is a 91, which is quite good since this is out of 100. The latter 2 scores are 2 different on-target metrics. You can find out a little bit more about them outside of the scope of this webinar, but if you click on the track name itself, you can read about exactly what the difference is between the score from the 2016 Dench paper or the Marino-Mateos score. So let's go ahead and move ahead with this particular guide RNA sequence. I can tell it's within the exon of beta-actin here, and it looks like the numbers are all pretty high, so this might have a good chance of working well. If I click on that guide RNA sequence, it brings me to a screen that gives me the entire guide RNA sequence, the NGG or PAM sequence, the specifics about the actual scoring algorithms for specificity and efficiency, and then, this is really nice, it shows you the number of mismatches, the count of off-target sites with a given number of mismatches. So I can see I have no direct copies of this exact 20 base sequence, and I'm also safe from 1 or 2 base mismatches. But as soon as we go to 3 mismatches, I have 14 particular sites that exist off-target. And then we start to see what those off-target results look like down below. Now, you can kind of just by looking at this, tell whether or not you think this is likely enough to prevent an off-target editing event. And in general, having 2 or more mismatches has a pretty high likelihood of disrupting or at least severely limiting the potential for Cas9 to cut off target, particularly if you're using a ribonucleoprotein approach, delivering Cas9 as a protein. So this looks like a pretty good guide RNA, and I'll go ahead and just copy that, and we'll order this in just a moment here. So we'll say this is the beta-actin guide RNA 1, and we'll save that. So now let's take a look at the MIT tool. And this is simply crispr.mit.edu. And this is just another nice alternative where you simply enter a search name. We'll just put example for now. And then you choose your sequence type, whether it's a region not distinct to the genome or a unique genomic region for any of these given species. So we've got a lot of options here. And then after that, you simply paste in the sequence. One limitation to this tool is it does only allow 250 bases to be submitted. And it takes a little bit of time for it to actually do an alignment test of all of the potential CRISPR sites within that sequence. So rather than waiting for those results, I've actually pre-populated some here so that we can take a look at what that looks like after you've submitted a job to this site. So here we have a graphical display of the sequence that I've submitted, and then arrows showing which direction each of the potential guide RNAs may be. And this one's split by which strand the particular guide RNA is on. Typically for the wild-type Cas9, it doesn't matter which strand you target. So in both cases, it'll induce a double-strand break. So any of these options would be good. And then I'm actually going to take a look here at the specificity scores. This is exactly the same algorithm as what we saw on the genome browser. And in this case, the highest score is an 87. And as you move your cursor over these results, you can also see the different off-target sites so that you can very quickly get an idea of how likely they are to actually cut at an unwanted site. In this case, the highest risk guide RNA off-target site has 4 mismatches at these 4 positions. It also will tell you if you have a likelihood of actually hitting a coding sequence and what that specific gene would be. So lots of great off-target information here. You can see that while this one's quite good at 87, if we take a look at this lower ranking score, only 2 mismatches exist for this first potential high-risk spot. So that hopefully gives you some sense of how you can use either of these tools to quickly choose a guide RNA. Let's go ahead and copy this guide RNA sequence, and we can use that as our second example. We'll say test guide RNA 2. So now we have 2 different guides we've designed. To actually order these, it's quite straightforward. You just hop over to the IDT website, choose the order menu, and then we have our genome editing section on the right-hand side. So I'll choose CRISPR-Cas9. We do also offer the CPF1 CRISPR system. That's outside of the scope of today's webinar, but we do have a recent webinar that discusses the CPF1 system in greater detail. So clicking on the Cas9 link, we can scroll down to the ordering section. And what we're ordering now is the actual CRISPR RNA or CRRNA. And that's the shorter of the two RNAs that actually directs the Cas9 nuclease on where to go. So since these are new RNAs that we haven't tried before, I'm going to order on the 2 nanomole scale. I simply click Order here. And then this should look familiar if you've ordered DNA primers from IDT before. So we'll copy the sequence name, we'll copy the sequence, and we'll go ahead and order both of these. So that's all there is to it. So I'm putting in a 20-base DNA sequence, not including that NGG PAM site. This is the only part of the sequence that you need. And then when I click Continue and eventually add these to the shopping cart, it will automatically convert them to the longer RNA format. So it's pretty easy to go straight from a design tool to ordering. In terms of the other components to the experiment, the CRRNA forms an RNA duplex with a tracer RNA. And that tracer RNA is a universal RNA. So you can use one tracer for all of your CRISPR experiments. So in this case, with this new project coming up, I'll just order one tracer RNA. There's 5 nanomoles of this, which is plenty to go with the 2 nanomoles of each of those CRs I'm beginning with. And then I'll need some Cas9 nuclease as well. So I'll order a small Cas9 nuclease. And for now, we'll go ahead and start with ordering those. And I simply add them to the order. And so that's it. We see that these now are in the correct annotation, and we have everything that we need. So I would just move on to checking out. And I have some other ones in my cart here. So that's how to go ahead and go through the ordering. We'll hop back over to the presentation here. And now that we have our guide RNAs, we'll place that order and that will be on the way. We'll look at assembling the ribonucleoprotein complex. So this is really actually quite straightforward. The 2 RNAs arrive in standard tubes like you're used to if you've worked with our primers before. It's dried RNA in a tube. Those are resuspended just by adding either a TE buffer or a duplexing buffer to that tube. The tracer RNA actually comes with the duplex buffer already. So if you don't have a buffer at the lab you want to use, that will actually come with the buffer that you can use to resuspend both oligonucleotides. Once they've been resuspended to the same concentration, you can mix them at a 1-to-1 molar ratio. And then they simply go through a quick heating and cooling step to release any secondary structure and allow the CRRNA and the tracer RNA to form that intended duplex structure. So then you have your proper guide RNA, and you again mix that guide RNA duplex with Cas9 nuclease at a 1-to-1 ratio. And these form that ribonucleoprotein complex very readily, so that these steps don't take long at all. And at that point you have your active RNP, and you're ready to move on to delivery through liposuction, electroporation, or microinjection. Altogether, these steps typically take less than an hour, so it's a pretty quick experiment. So now we're ready to actually deliver the RNP to the cells or to the samples that you're working with. So I'll talk about the differences between these 3 common delivery methods. So with liposuction, this is probably the easiest of the delivery methods because you don't need an instrument to even get started. You simply order the lipids. We work with and recommend RNAi-Max, lipofectamine RNAi-Max, and have found CRISPR-Max to also work very well. It does work for a number of cell types, but unfortunately it's not compatible with a full range of cell types. So some more challenging cells we'll talk about with electroporation. One nice thing about liposuction is that you need very small amounts of the actual RNP. So even the smallest scale products that we offer as the CRRNA and Cas9 and tracer RNA will last for many experiments, sometimes even over 100 experiments from our smallest scale options. So it can be a very high throughput way to run and test a lot of experiments, especially if you're working in a 96-well plate and you can assess these quickly. It makes it a very inexpensive way to do a lot of CRISPR testing all at once. As I mentioned, not all cell types are compatible with liposuction, and some cells start to show toxicity when the lipid level is too high. When that happens, generally we need to move on to electroporation, and this is much more compatible with challenging cell types like primary cells or induced pluripotent stem cells. We've used electroporation and collaborated with other users on electroporation to deliver the ALTAR RNP system to a very wide range of cell types, including primary cells and iPSCs. So this can work very well. The only kind of caveat to electroporation is that up front it does take some optimization effort to make sure you have all the conditions just right to work well for those cells so that you have good delivery and high viability of those cells. The nice thing about some electroporation systems, particularly the nucleofection system from Lonza, is that it does offer a high throughput option, so you can actually electroporate an entire plate at once. It does require higher amounts of the ribonucleoprotein complex, though. So you can't stretch your reagents as far with electroporation, but it still works very well with these challenging cell types. Of course, you have to have the electroporator to begin with, so that initial investment exists unless you have hopefully a lab nearby that might let you borrow their instrument for the day. And the consumables can start to add up as well. So those are some of the cons to the electroporation option. Lastly, for microinjection, this is, of course, much more model organism related. So if you're interested in making a new mouse strain or you're microinjecting into zebrafish or C. elegans, this is typically what's used. One great thing about microinjection is that you know exactly where the ribonucleoprotein is being delivered. You're not relying upon the protein to find its way into the nucleus, but rather you can actually microinject directly into the nucleus and know that that genomic DNA is accessible to the protein. Because of that, you can typically use a lower ribonucleoprotein amount. So again, this probably isn't as low as liposuction, but you can have a lot of injections out of a small amount of ribonucleoprotein. With microinjection, it does typically require very good hands or experience having done many microinjections in the past. And you need the equipment to actually do the microinjections. So sometimes this can be a more expensive method as well. To go into further detail, I think would take up a little bit too much time, but we do have, and I wanted to point out, some very detailed guides for really walking through on a step-by-step basis, delivering via liposuction and electroporation to different cell types. These are available on the IDT CRISPR-Cas9 website, as well as for the CPF1 system. And something that has been really great is we've actually been able to share some user-submitted protocols where we haven't done any model organism work here in our R&D group, but we've had collaborators that have decided to share their protocols with the wider community using these reagents to deliver into mice and zebrafish and C. elegans. So we have a new protocol coming soon for actual mouse electroporation, mouse zygote electroporation. And if you use these tools and find a very efficient method that you'd like to share with the community, please feel free to send that our way. We'd love to share it, and I think that helps everybody accomplish better research. So now we've successfully delivered these to cells, and we'll quickly talk about collecting genomic DNA and then moving on to analysis of those results of the CRISPR editing. Collecting DNA is pretty straightforward. I'm guessing many of you have already done this. This is detailed much more specifically in the protocol, but in short, you have a couple PBS washes of your cells, lyse the cells. We use the quick extract solution, and we transfer them to a new container and vortex that material before heating them and doing a final dilution and spin down step. So at that point, you're ready to go with your genomic DNA, and you can move on to analyzing those samples and looking for those CRISPR edits. And the way that we do that is using an assay called the T7 endonuclease 1 assay. So this enzyme recognizes mismatches in double-stranded DNA. And to take advantage of this, we begin with a PCR amplification step, designing primers that flank the CRISPR target site that you're interested in. So in the example on the left here, if the CRISPR target site's right in the middle of this segment of DNA, amplifying across that region, if CRISPR editing has occurred, you'll end up with some wild-type DNA that wasn't edited, or it was edited and repaired successfully without errors, or you might end up with some insertions or some deletion events. After the first initial rounds of PCR, there's one final heating and cooling step, which allows all of these different strands to denature and then reform, potentially introducing heteroduplexes right at that CRISPR target site. So if a wild-type strand binds to an insertion strand or a deletion strand, you end up with predictable mismatches at that target site. And those are the substrates for the T7 endonuclease 1 enzyme to cleave that double-stranded DNA, providing you with digestion products that can be run out on a gel, capillary electrophoresis system, or fragment analyzer. And you should have predictable products where you can see a full-length band and then compare it to the amount of the digested products to have an idea of how much editing has occurred in those samples. So this is a very simple assay. It doesn't require a lot of extra steps other than PCR and the digestion with the T7 endonuclease 1. And it's something that can really be done in high throughput. So we use this routinely to have very fast results to confirm CRISPR editing has occurred. The downside that I'll point out to this assay is that this enzyme doesn't detect single base insertion deletion events. So it does actually underrepresent the overall level of editing that occurs in those cells. But it still gives you a very good relative idea of how much editing has occurred between different guide RNAs that you're testing, and gives you enough of an idea of whether or not you can move on to the next steps you're interested in the experiment. To be absolutely sure of what those edits are, typically Sanger sequencing or next generation sequencing is still the best method to confirm exactly what the products of those edited cells will be. So we'll spend the last few minutes here talking about homology-directed repair. And just as a reminder, this is actually introducing a single or double-stranded DNA product to actually create a specific change at that CRISPR cut site, so that when the repair event happens through homology-directed repair, you end up with a new mutation at that site, assuming that you have high levels of HDR occurring. So from a preparation standpoint, this looks very similar to what we saw for the initial RNP preparation. The only difference is that now you'd introduce your HDR template and the final step along with the delivery of the ribonucleoprotein complex. So ultramer oligonucleotides, these are our higher fidelity oligosynthesis products, are commonly used as HDR templates, and we'll talk about why that is. But in terms of a workflow, it doesn't change things very much. You're simply adding in this extra oligonucleotide to that last delivery step in your experiment. So some considerations to keep in mind to begin with for HDR. The size of the mutation you're trying to create really dictates what type of template you should use. So for point mutations and smaller sized insertions or tags, I highly recommend using single-stranded oligos. And for the highest fidelity, the ultramer oligos that IDT offers are a very common choice. Standard desalting tends to be sufficient for purification, and the overall unit length for ultramers allows up to 200 bases. So we typically recommend for these smaller insertions homology arms, which we'll talk about in a minute, that match either side of the cut site, ranging between 30 to 60 nucleotides. So that means you can easily fit a approximately 100 base insertion, and sometimes even a little bit larger, within the length restrictions for an ultramer oligonucleotide. Or you might just be simply making a single point mutation, and that's just fine too. For even larger insertions, though, if you're moving beyond, say, 150 or larger bases, typically double-stranded donor material is used. And the reason for that is not only the size limitations for ultramer oligonucleotides, but we typically have to move to double-stranded DNA for those really large inserts. And when we move to double-stranded DNA, it seems that longer homology arms are needed to have high rates of HDR, typically a homology arm greater than or up to or greater than 500 bases in length. So either using a donor plasmid that's generated or something like a G-blocks gene fragments as the donor template are typically used. The last consideration, it refers to positioning. So wherever your desired mutation site will be, ideally you'll choose a CRISPR guide RNA site that's as close to that mutation as possible. And then in addition, you'll design your HDR template so that it really sits on top of that mutation and CRISPR cut site. That seems to really drive the HDR rates as high as they could potentially be for that experiment. And we'll look at some data too, so that you can have some sense of what you can expect. Of course, that's very cell type and experiment dependent as well. So this was a survey experiment where HDR templates centered on a CRISPR target site were tested with varying arm lengths, as shown on the left. So from just under 30 bases to slightly over 90 bases. And these are centered on the CRISPR cleavage site, and the insertion element here is actually a restriction enzyme site. So an EcoR1 site was put in with, on either side of the insertion site, sequence that matches the sequence of the wild-type DNA on either side of where that CRISPR RNP will cleave. And we'll take a look at very specific HDR template design in just a couple slides. So the goal here was to try to assess HDR by looking at whether or not cleavage through this insertion of a restriction enzyme site occurs. This particular experiment used a guide RNA targeting EMX1 in HEC293 cells, and it was done via liposuction of the RNP into those cells. The ultramer oligos were standard desalt, and they were delivered at a 3 nanomolar concentration, either as double-stranded DNA or single-stranded DNA from either strand of that template. So after 48 hours after delivering these to the cells, the genomic DNA was isolated, PCR amplified, and then tested either through the T7 endonuclease 1 digestion that I described earlier, or through EcoR1 digestion to determine how much editing overall had occurred through non-homologous end joining, and how much of the editing occurred through homology-directed repair, where that EcoR1 site was now present after HDR had happened. So here's the data from that experiment, and what we see is in blue, this targeting strand, so the strand that the that the RNP actually binds to. In orange, the non-targeting strand, and then in green, the double-stranded DNA template. On the top, shown by the points, are the level of editing based on the T7 endonuclease 1 assay, so we expect this to be higher. And then the rates of homology-directed repair are indicated by the bars. So we range from kind of mid to high 30s across the board. Now, in this experiment and in additional experiments that we've run, we find that overall a 30 to 60 nucleotide homology arm length seems to provide quite good and consistent results. So that tends to be the range that we work with now for our ongoing HDR studies. When you move to longer insertions, I think that the homology arm length may vary at that point. But as a general rule, this is a good guideline to start with when you're designing homology-directed repair templates for simple and small insertions. Now, one thing I'll mention about this is you'll notice that the double-stranded DNA EcoR1 insertion seems to be quite high, almost on par with the single-stranded DNA. And it might be tempting to consider using a double-stranded DNA template, but what we actually found was occurring in this case is that this is artificially high because of the type of incorporation that's happening with the double-stranded template. And if we take a look at the fragment analysis results of both the single-stranded template and the double-stranded HDR template, what we see is with the single-stranded template, we have our wild-type product, the full-length amplicon, shown in the tall red bar here, and then we have the two cleavage products after the EcoR1 digest. So as predicted, we have two cleavage products, and we're seeing this type of HDR occur, where we have just insertion of the desired restriction enzyme site. With the double-stranded DNA, however, we actually have a duplication of the digestion products. We have our expected HDR event, which matches the peaks for the single-stranded template, but we also have these larger products that are the result of actually having a blunt-ended insertion of this double-stranded DNA product. So we actually have the non-homologous end joining process being used to essentially force this double-stranded donor into the cut site. So we have a replication of those homology arms. So this is, of course, not what you want in most cases in the experiment. So when you can, single-stranded templates tend to avoid this issue entirely. We haven't ever seen this type of product with a single-stranded HDR donor. Another interesting thing is, as you test various sites and attempt to achieve HDR, the actual rate of HDR can really vary quite widely across different sites, even when keeping all other conditions the same. So here's just an example of that shown with 5 sites where some of them have really terrific HDR rates, where we're seeing even up to 50% HDR in these cells. But in other sites, we just see not very high levels of HDR occurring. So this tends to be very specific to both a guide RNA, the gene where you might be targeting, and might even be specific to the specific HDR template. So sometimes you do need to test more than one site, and we'll take a look at designing the actual HDR guides here. So taking a look on a sequence level of what an HDR repair template will look like. On the top, we have a wild-type sequence. And in this example, we're looking at changing this particular codon into a stop codon to try to disrupt this particular sequence from being translated successfully. So we're making 2 changes, shown in red here. And the way that we'd want to get started is to first find all of the PAM sites, all of the NGGs in this sequence. So we can see we have 4 particular PAM sites. And in this example, we'll pick the closest one to where we're trying to introduce the actual mutation. So that gives us this particular protospacer sequence just upstream of that PAM site. And as I mentioned before, the Cas9 nuclease will cut between bases 17 and 18 of that protospacer. So here's where we'll actually cut, and now we just need to design the HDR template. And the thing that makes this really easy is the HDR template should simply be the desired mutation in the sequence that you'd like to have at the end. So in this case, I've can quite literally copy the sequence from what I want as my desired end product. And in this case, order this as a single-stranded oligonucleotide with 30 to 60 bases upstream and 30 to 60 bases downstream, and my desired mutation. I'm showing just one strand here, but both strands could be tested. And as we saw from site to site, sometimes the sense or the antisense strand will work better. One thing to note, though, and one last consideration on designing that HDR template, is it's important to think about after that HDR has occurred, whether or not your guide RNA sequence has a risk of recutting at that site that you've mutated. Now, in this case, we have two mutations that have been introduced, and they're very close to the actual PAM site itself. So that should be quite disruptive, but it's not a guarantee that this won't be cut again, even with these mismatches here. So if you're concerned about that, you can always introduce other silent mutations either within the protospacer region itself or even within the PAM sequence. So that's something to consider, how your final HDR template will affect recutting events. To take a look at an example with a larger insert, in this case, we're looking at putting in 33 bases between the two bases in red here of the wild type sequence. So our desired edit looks like this. We're putting in 33 bases in the middle. And again, we'd find all of the PAM sequences, still choose the closest one in this example. You could always test a couple of them. And here's where we're going to cut. It's not right on top of the sequence, but that's still okay. And then our HDR template will, again, look like our finished product that we want to have. 30 to 60 bases, to 33 nucleotide insert, and then 30 to 60 bases downstream. In this example, we have much more protection against recutting events because it's very unlikely that this insert will match these final 5 bases of the protospacer. And then again, it may not even have a NGG PAM site in this example. So this one wouldn't be so much of a worry, but it is something I like to point out to make sure that you can avoid that risk of recutting events. In terms of synthesis options for HDR templates, you could use a standard oligonucleotide, just like you're ordering a primer. And for IDT, that's offered up to 100 bases in length. The ultramer oligonucleotides, it's a slower synthesis process, so we have a higher fidelity, and those allow up to 200 bases in length. And one thing I'm excited to briefly mention, this is an unreleased product, but we are planning to actually start offering very long single-stranded DNA fragments that will actually go up to 2,000 bases in length. So this is in an early beta testing phase, but if that's something that you're interested in, and the ultramer oligonucleotides are a length restriction for you, I do have a sign-up URL here that you can either write down now or collect from the slides later. And that sign-up will allow you to get information as we move further towards releasing that product. So in summary, CRISPR guide RNA design has really become simple with a lot of free online tools. I don't have a specific recommendation for which tool you should use, but I do recommend understanding how those tools work, what the rankings are based upon, and whether or not they relate well to the type of experiment that you're doing. Of course, efficient ribonucleoprotein delivery can be achieved through any of the delivery methods that we discussed, and there are optimized protocols for all of these and many applications on the IDT website. And then if you do decide to try homology-directed repair, it's a great way to introduce simple mutations and insertions at your target CRISPR site. The repair templates are pretty easy to design, and they're synthesized quickly for your experiment. I want to point out that our research and development team is really responsible for developing the tools for the Alter system that we have today. They've worked very hard on optimizing this system and making the best guide RNA system that we could. It's optimized both in length, it's chemically modified, and we really think that it's a really great way to go. So hopefully you'll give it a try, and I really appreciate their efforts. Some additional resources and support are available online. So we have protocols, we have the user methods that I described, and some frequently asked questions. Just visit our website and go to the CRISPR-Cas9 section and check out that support tab. If you have questions that aren't addressed there, we have a really great scientific application support group. And these are scientists from the bench that have actually a lot of experience with a lot of our tools that we offer, and they can provide suggestions on how to really optimize your experiments. You can also visit us at idtdna.com/contact us for region-specific phone contacts if you'd rather give us a call. And with that, I really appreciate everybody's time and attention. It's my pleasure to be able to give this webinar today, and I'm happy to answer any questions.
All right, so thank you very much, Justin, for that presentation. If you haven't already asked your questions, still type it into the questions box at the right-hand side of your screen. We've already got some of those. We're not going to make it through all of them in this portion, but we can respond to you individually by email as well, so you will get an answer. I want to direct you quickly to the chat box. There's a link there to both the slides and to the beta testing site for the longer single-stranded DNA that Justin had mentioned. So we'll squeeze in a few questions here. I know we're at the top of the hour. If you do need to go, that's fine. Like I said, we'll be following up with you by email, and you'll get links to the webinar recording and the slide deck. All right, so let's just get started here, Justin. There were a couple of questions actually about stability of the guide RNAs and the ribonucleoprotein complex itself. So the two RNAs come together at that short double-stranded region between the two of them, and you anneal those. And there were questions about how stable is that when you go to transfect it or electroporate that. And then also, is that more stable when it's incorporated into the ribonucleoprotein complex? Do you have any comments on that?
Sure. Yeah, that's a really great question. So there are that's correct. There's a short region of homology between the CRRNA and the tracer RNA that readily forms a duplex as long as there's sufficient salt content present in that buffer. So that's why we recommend resuspending both of those RNAs in either a TE buffer or what we use is a duplexing buffer. It's a HEAPS potassium acetate buffer. And with that sufficient salt content, those two that interaction is thermodynamically favorable. And then it is actually further stabilized once it has complexed with the Cas9 protein. So that complete Cas9 RNP, once that complex is prepared, it's actually quite stable. And we've actually done a number of tests with freeze-thaw cycles and dilution of these RNPs and found that it can actually last for quite a while. So that's one convenient thing for preparing your experiments is you can actually prepare the full RNP and keep it in the refrigerator for easily up to a week at a time so that you don't have to worry about scrambling on the day of an experiment to get all these things ready. If you want to store them longer term, we actually recommend once the protein is diluted to store it at -80 so that it freezes very quickly, but it does remain active after freezing. The only thing we wouldn't recommend in terms of long-term stability is putting it at -20 without that glycerol content present, just because we don't want it to freeze slowly and risk deforming or disrupting the protein itself.
Okay, you actually jumped ahead to my other question about storing the RNP, so that was great.
Okay.
The next thing that I have here for you, Justin, is a couple of questions about the design tools that you showed. So in the MIT design tool, do you know why there are two different zebrafish genome options? There is there's one that says Dan Rear, so Danio Ririo.
Yeah.
And then there's another one that's a GRCZ10.
Yeah, so those are just different builds of the actual genome for the zebrafish assembly. So the Danio Ririo 7 is an earlier assembly, and I believe the reference genome 10 is the most recent zebrafish build. I'm a little rusty on that specific species, but I think that that's just a difference between which reference assembly is going to be scanned when it's looking for those off-target effects.
Okay. And then this is an interesting question, working backwards a little bit. If somebody finds a published guide RNA and they want to come back and see like what off-target tools something like this might predict, is there a way to do that?
Yeah, that's a great question, and actually I really like that you can do that with the MIT tool. So the one thing that you will need, and I've been able to do this by putting in that 20 base sequence and the PAM itself. So you'll need the NGG or whatever the N is. So if you have your 20 base sequence, so let's say we put this one back in here, and then you know what the NGG is. I can't remember if it was CGG here. But this should be enough where you could actually submit this, choose the correct target genome, and it will look at this sequence, identify that there's should only be, yes, only one, it can only fit one particular target within here, and it should still give you that same output. I'll let this.
That was actually a pretty cool question. I actually did not know the answer to that.
Yeah, so I don't know if this will run fast enough, but we should be able to see that in action here.
In the meantime, so we're not just watching that tick away for a couple of minutes, let's move on to the next thing. Again, regarding the MIT tool, does the MIT score for the guide RNA only mean less off-target effects, or is there any prediction in there about the efficiency that somebody might expect from that guide RNA?
Yeah, that's correct. So this tool does only show you off-target rankings. It doesn't provide any kind of on-target analysis. On-target is in particular the one that I think is sensitive to how that on-target ranking was built. And so I think that that's where more variability comes into play. Largely from what we have seen, taking a look at the research that has come out of IDT's R&D group, is that it's quite challenging to successfully predict how efficient a guide RNA sequence will be. There are a lot of guide RNAs that I've seen the tools predict to be bad that end up being good, and some that are predicted to be good that are bad. And so I often, when doing my own designs, tend to more heavily weight the off-target risk, but that's really a personal experimental decision. I don't want to tell anyone how to do that too much, but I wouldn't always rule out an experiment or a guide RNA because in the on-target efficiency appears to be low. It might still prove to be an effective site.
Okay. All right, the next question I have, I've got a couple of delivery questions here for you. So for homology-directed repair, you have your HDR template. If you add that to the transfection or electroporation or whatever of your ribonucleoprotein or guide RNA, Cas9, whatever form you've chosen to do that in, does that extra molecule impact the effectiveness of the transfection itself?
That's a really interesting question, and what we've seen is for electroporation delivery in particular, the presence of that extra DNA molecule can actually help improve the rate of delivery. So we've actually created, and I didn't mention this during the presentation today, but we actually offer a separate specific carrier DNA molecule called the electroporation enhancer for CRISPR-Cas9 and for CPF1. And this is actually a long single-stranded DNA that was designed to avoid homology with human mouse and rat genomes, and its only purpose is to actually help improve the rate at which that ribonucleoprotein complex gets into the cell via electroporation. So a homology-directed repair template can actually act much in the same way, except here it actually has a secondary purpose of also mediating homology-directed repair.
Sure. This next question is interesting. I've seen publications that talk about things like this, but I don't know if we have any experience with this. If you want to make 2 cuts in your genomic sequence at 2 different PAM sites. How far apart do those 2 sites need to be in order for there to be space for the RNP to act on both of them?
Yeah, that's a really good question, and that's something that we've started exploring a little bit as we've looked into multiplexing ribonucleoprotein. It does seem like the footprint of the Cas9 molecule needs a sufficient amount of space to be able to make 2 proximal cuts, and it also depends upon whether or not the 2 guide RNAs are on the same strand or on opposing strands. So if we think about the worst case scenario where it's 2 guide RNAs on the same strand, and so the orientation of the both RNPs are on the same strand, I would recommend at least 20, if not moving to maybe 30 or more bases apart from one another. We're still studying that, and I can hopefully have that included in some future online material, but it does seem like there is, I'd say, a minimum of about a 20 base space necessity to make sure that both RNP molecules can fit into that space and induce the break.
Okay. The next question that I have is how long is the Cas9 active inside the cells after you do the electroporation?
That's a good question, and it's going to depend a bit upon the cell type. We have done some testing and tried to kind of track the Cas9 after delivery. It seems like for cells that have high turnover, typically Cas9 stops being detectable by Western by around between 3 to 7 days. That's kind of a long time frame. Of course, it's very cell type dependent, but in terms of lifespan for delivering reagents, it's pretty short. Most of the editing happens within 24 or even 48 hours. We see a plateau of the actual editing events, and the Cas9 lasts comfortably that long and not much longer. So it's delivering Cas9 as a ribonucleoprotein complex is really the perfect solution in that it's there long enough to do what it needs to do, but not enough time to kind of cause trouble and stick around for too long. And that's really why we don't prefer the DNA delivery approach, where you end up with Cas9 sticking around longer than you'd like.
Okay. So one more question for you. Is there a good method for suppressing the mechanism for non-homologous end joining in order to increase homology-directed repair efficiency?
There are a lot of different methods that have been discussed in the literature. The most common of which is probably the SCR7 small molecule, which I'm trying to think of it, it might be a ligase inhibitor, but the verdict from what I've seen is still out on whether or not there's one real runaway winner at suppressing a non-homologous end joining. A lot of things have been tested, and that's certainly something that we're interested in to help boost the rates of HDR. But it's not something that we've have a clear answer on yet. So I think there are certainly some options out there. I think we haven't yet determined what other negative impacts or other risks might come along with some of those non-homologous end joining inhibitors. But yes, things certainly exist, and the most common thing is looking for small molecules that inhibit NHEJ.
Okay. So we still have quite a few questions to go, and we are about 10 minutes past our time, and I see quite a few people are leaving now. So if we haven't responded to your question, we still will respond, and I really appreciate you participating in the discussion and asking these great questions. So you'll definitely be hearing from us. We'll also be sending you a link to the recorded webinar. Like I said, the links to the slides and to that beta testing site for the longer DNA, single-stranded DNA, are in the chat box right now, but we'll send you links to stuff also in that email, so you can have access to that later. You don't have to grab everything now. So with that being said, this was really great. Justin, thank you very much. Thanks everyone for participating. We definitely will be coming back in the near future with more CRISPR webinars as well as other molecular biology topics. I think certainly we'll be talking more about homology-directed repair in the future, especially with those longer single-stranded pieces of DNA coming in the future. So look forward to those things. Anything else, Justin?
Nope, that's it. It was a pleasure, and thanks everyone for joining the webinar today.
Thank you, everyone, very much. Have a good day.